my fellow lifelong curious people. It's time for our main event, our first paper together as a book club. Together, we're going to explore the breadth of awesome academics going on out there right now. Whether you read academic articles regularly or just want to see what's going on out there, I want to help you to know about and understand what these experts are working on, and how to look critically at what you're reading, in the original form of the word critically, to make sure that it makes sense and to figure out how much you can trust it. I also highly encourage you to fact check me. I always double check what I say, and I post the sources that I use for the self-fact check in the description, but I want to know if I miss anything. One of the most basic parts of science is to rethink even your most basic knowledge if real facts contradict it. If I make an error, I want the comments of that video to explode with corrections. I'm counting on you. This series' academic article is titled Potent by Design, Amino Acids Mimicking Porous Nanotherapeutics with Intrinsic Anti-Cancer Targeting Properties by Wu et al. If you haven't read it yet, the article is free at the link in the description. If you'd like some background before getting into the nitty-gritty of the paper, please watch my Cancer 101 video and Nanoparticles 101 video. This will give you some basis for what's going on in this article, even if you're not a biologist. I'm going to present this paper in this video and ask a few questions of you, but my real hope is for the magic to happen in the comments. Please post thoughts, questions, and extensions. We'll have a party. Without further ado, welcome to the Academic Article Book Club. The first thing one wants to do when delving into an academic article is figure out what that title means. Paper titles are basically the abstract of the abstract, which is why they're often so long and descriptive. Potent by Design is pretty self-explanatory, but basically they used molecules that mimic amino acids, the building blocks of proteins, on porous nanotherapeutics, which are the delivery mechanism, to deliver a payload of anti-cancer substances. Using the amino acids to coat the nanoparticles gives them intrinsic anti-cancer targeting properties. For those that don't read these often, the abstract is basically the whole paper in a nutshell. It has a little bit of background, general methods, basic results, and a little bit of what that means. It's basically the elevator pitch of the paper. It's not really possible to summarize an abstract since it's basically already a super tight summary, but I'll do my best. Cancer growth is highly dependent on external sources of various resources, including and especially the building blocks of proteins, amino acids. Cancer cells will greedily take in as much as possible, so they exploited this by covering a very toxic payload in yummy-looking amino acid mimics. They tested 9 amino acids that are only introduced into cells through nutrition, also known as essential amino acids, to see which one will be uptaken the most and release its payload the best in cancer cells without hurting non-cancer cells. The one that's shown above the others is the one functionalized with L-phenylalanine. It murdered the cancer cells at a lower concentration than the others, and it left the non-cancer cells mostly alone. They suggested a possible mechanism for this might be that cancers overexpress the large neutral amino acid transporter called LAT1. This phenylalanine functionalized nanoparticle carrying a toxic payload activates not one, but both of the possible pathways that lead to apoptosis or programmed cell death, exerting a, and I quote, potent double whammy anti-cancer effect. I love these authors. This paper was fun to read. Once they'd determined efficacy, they tested it on a small number of SCID mice, which are mice that are modified to be immunologically deficient so that they're not testing the immune systems of the mice, but the effectiveness of the treatment. Without anything other than this treatment, including anti-cancer drugs currently on the market, they suppressed tumor growth by 60%. One thing that cancer cells do well is recruit resources. Often, they'll make extra transporters to bring in extra nutrients. And there are studies of angiogenesis, or the ability of some cancer cells to create or recruit blood vessels to bring nutrients directly to the tumor. This group used the fact that cancer cells make a crap ton of amino acid transporters to swipe amino acids from the surrounding tissues to design a Trojan horse. No, really, they called it a Trojan horse. As I said in my How to Read an Academic Article video, all academic articles are going to try to be as precise in their language as possible, but not all of them devolve into pure jargon, and some of them are quite delightful to read. Anywho, this Trojan horse is made by covering the delivery particle in amino acids. We'll go over how they figured out which amino acid to use in the methods and results section. Many current cancer therapies involve the patient stopping their amino acid intake. Together with the non-compliance in patients to the strict dietary requirement, the clinical adoption of a nutrient-based approach to curb cancer growth by limiting amino acids intake is significantly curtailed. If I am dying of cancer and my doctor suggests that on top of the chemo or radiation or whatever's making me miserable and nauseous, that I then stop eating when I feel good enough to stomach it, I'm gonna eat their head off, and that won't help anyone, now will it? So, figure 1 is illustrating this difference. The top is the general current strategy where you starve the patient of amino acids. Since the cancer cells are dividing and growing much faster than normal cells, they'll feel the effects of the lack of ability to make proteins much faster than normal cells and become unstable and apoptotic, or programmed for cell death. If you can get the cancer cells to all starve before the patient does, you win! I say it in a sarcastic manner, but controlled fasting can trigger apoptosis in weaker cells throughout the body, especially cancer cells. I'll link in the description to the Found My Fitness topic on fasting. Dr. Patrick links to tons of studies on the topic. Just be careful, okay? While there are good studies on the benefits of various types of fasting, eating disorders are a real and dangerous thing. Things that can be good for your body in controlled sessions for a few days at a time may not be so good if your brain, which is part of your body, I might add, is sick. I'm also linking to some sources that may help if you or a loved one is struggling with an eating disorder. So, back to the picture. The proposed method is on the bottom. Instead of restricting the patient's diet, you cloak a nanoparticle in a delicious amino acid disguise. The patient consumes it, and then the cancer cells selectively eat it because it just looks so yummy. The nanoparticle releases its reactive oxygen species inducing core. At a high enough level, the ROS will stress out the cell enough to induce apoptosis. Nanoparticles are the delivery method used. Throughout the article, they called them NanoPAM, which stands for nanoscopic porous amino acid mimics. Literally nanoparticles that are made in such a way to give them small pores all over the place that are covered in amino acid mimics. Still, whenever I read NanoPAM, my mind just went,
It's him! Careful, it's hot!
For substances that have poor bioavailability, poor solubility, or that just won't get into the right cells for whatever reason if it's left out in the open, nanoparticles are the way to go. They surround whatever you want them to surround. In many cases, life-saving drugs, and in this case, a nasty toxic substance that will poison the cancer cells in a high enough concentration. And then you can modify the outside to change what it'll do in your body or where it'll go. Go watch my whole video on nanoparticles attached here for your viewing pleasure. For this study, they used a mesoporous silica nanoparticle made of silicon dioxide, which is silica gel. What part of do not eat do you not understand? Nah, as a nanoparticle, it dissolves in water and surrounds the payload. The reason for not eating silica gel as those little balls is because it's a desiccant, which means that it absorbs water like crazy. This means that they're great for making sure a product doesn't get too moist in shipment, but if you, who are made of water, swallow one, then you will get very dehydrated very fast. And it may cause tissue damage from suddenly being drier than a desert in one little spot in your esophagus. Anyways, silicon dioxide as a particle that hasn't been dried out is given the generally recognized as safe label by the FDA. Silica is also very easily modifiable, leading to tons and tons of studies where you take your molecule and you attach it to a glass bead, glass is also silicon dioxide, to then pull on them to see how strong their bonds and folds are. I'll link to Dr. Perkins's lab page if you want to read more about that. Alrighty, so now we're gonna get into the nitty-gritty of the paper. How and why they did each step, and what the results mean towards what we talked about in the abstract. The first part is about making those modified nanoparticles to determine which amino acids, if any, work best against the cancer cells without hurting those normal cells. They synthesized amino-functionalized mesoporous nanoparticles, NH2-MSN, via a modified classical Sturbe method, which means they stuck some amine groups onto the nanoparticles using a version of the way to make the nanoparticles that ends with an amine group stuck on. Mesoporous means that they have pores that are mid-sized. Smaller holes, less than 2 nanometers, are considered microporous, while larger pores, greater than 50 nanometers, are considered macroporous. Sure, that makes sense to me. These pores affect the surface area and the ability to efficiently deliver their payload. They also help it be even more modifiable. I don't know too much about the physical characteristics that allow mesoporous nanoparticles to be the best for this purpose, but the Science Direct page on it assures me that it is especially great for cancer therapeutics. I'll link down below to that page. Functionalizing the particles with an amine group allows them to attach the amino acids, since there are several amino acids that already have an amine functional group. They also left some as just the naked amine attached to the nanoparticle, so they can see the difference in effect between a basic nanoparticle with the payload that has gone through the whole process, versus the amino acid covered nanoparticles with the payload. This also tests to make sure that the FDA's generally recognized as safe label is correct in this usage as well. They then checked to make sure that everything did what they wanted it to by looking at it under an electron microscope. They saw wormhole-like mesopores of between 2 to 3 nanometers, which is the smallest that they can be to still be considered a mesopore by definition, throughout the approximately 30 nanometer particles. I've already linked to my nanoparticle video, but there I talk about the importance of particle size on uptake versus payload. I was able to look up what a mesopore was when first reading this, but when I tried to look up wormhole-like mesopore, it just kept giving me more scientific articles that didn't really define it clearly. My coworker who has worked with nanochemistry before said that it might be that they go all the way through the shell, or possibly that they have a snaking pathway through the shell. I was under the impression that pores by definition go all the way through the shell. If anybody watching this knows what makes a pore wormhole-like, or if it's the snaky pathway one, please let me know. I love the name and wish to know! They then checked to make sure that the amino acids were actually still acting like amino acids by doing an FTIR analysis. This is where you shine an infrared light onto the particles to excite them into intense jiggling. As they get down with their bad selves, they give off energy signatures that tell you what kind of structures you're looking at. You can watch the Khan Academy video that I linked in the description to learn more about what it looks like and how to read the data generated by an FTIR instrument. But yeah, for the naked amine particles, they had spikes at the points where amino groups and carbon-hydrogen bonds would be giving spikes. So that's a success! For the ones that have amino acids on them, the FTIR successfully got new peaks in all the places that correspond to the various bonds in the amino acids, as well as those that correspond to the new bonds between the amino acid and the silicon. The nanoparticles were then shot with light so that they could measure how much the light bent to determine how big the nanoparticle was, so that they could make sure that the nanoparticle was under 200 nanometers. Now that they've finished checking that they've made what they intended to make, an amino acid covered tiny silicon ball with contents that would wreak havoc on an unsuspecting cancer cell, they can get started on the actual study. So they fed their Trojan horses to highly invasive human breast cancer cells, aka cells that like to make big old tumors and suck up lots and lots of nutrients at the detriment of their organism. Also, using it on human cells like this means that when down the line we want to use it as a therapy on humans, there will be data saying that it does, in fact, work on human cells. On this chart, deep green means that everybody lived, and deep red means that everybody died, with various light greens, yellows, and oranges in between. The one that didn't have any amino acids on it is being used as a sort of method blank. It doesn't have the things that we're testing on it, but it has gone through the whole process up to that point. This way you have yet another check to make sure that you did in fact make it right, but also that you do in fact have to go through the whole trouble to put an actual amino acid on there. Luckily, the naked amine didn't do much, but did a little bit at higher concentrations. To me, this says that not as many of the particles are getting all the way into the cancer cells, which means that you need that higher concentration of reactive oxygen species inducers to get that same effect. It also says that the silicon dioxide isn't killing things on its own. They tested a bunch of amino acids that are all essential, meaning that the cancer cell doesn't make them on its own and will have to get them from the outside. They also all have that NH2 group on them, meaning that they can get stuck onto that NH2 on the silica ball. There is one little guy that sticks out as more effective than all the others. This yellow at the lowest concentration here means that it doesn't take much to make this guy super effective. This guy is phenylalanine, which is used in a whole bunch of proteins and is the precursor to several things like dopamine, adrenaline, and tyrosine, which is used in signaling cascades. The authors also noted that it looks like the ones that worked the best are also the most hydrophobic. Nanoppam, the PAM with the phenylalanine, killed a whopping 80% of the cancer cells at high concentration. Now that they know that phenylalanine will do the best cancer murder, they test my assertion that small nanoparticle is best nanoparticle. So yeah, you can see that 30 nanometer blue line get progressively more murderous at higher concentrations, with the 80 nanometer one following that same pattern but being less murderous on all counts. The 170 and 260 nanometer ones hang out at approximately the same rate if you look at the overlap of the error bars. They get a little bit more murderous at the high concentrations, but not much at all. The authors hypothesized that this is because the cancer cells will actually eat the nanoparticles via endocytosis. They tried inhibiting endocytosis by using sodium azide and also by making it really cold. Both results say endocytosis likely. So they moved on to a more specific mechanism of LAT1, which is a transporter that specializes in valine, leucine, isoleucine, and phenylalanine. When they used BCH, which is a LAT1 specific inhibitor, the nanoppam couldn't get its murder on much at all. You can see that there's a little dip from the control group, but not much. They looked at LAT1 expression in various cancerous and non-cancerous cells, and you can see that with a little bit of overlap in error bars, the cancer cells have significantly more LAT1 expression than the non-cancer cells. Once they prove this and they can see the various amounts of the transporter, it's back to murder time. Their control was still mostly alive at the end, which is good. That makes it good to see the nanoppam's results. At high concentration, the cancer cells were all 50% or more dead, with the stomach cancer and the high LAT1 expressing breast cancer both in the red range. Meanwhile, the most affected non-cancer was the immortal skin cell Hackat line, which was in the yellow-green range, so that means that non-cancer cells were generally left alone. Cisplatin, a chemotherapy drug, is their positive control because it kills pretty indiscriminately. Positive controls tell you if you're doing your method right, because if you are, then those positive controls will do the thing you're testing for a lot every time. Yikes, that is some red going on in those non-cancer cells. They then moved on to look at the action of the payload. It stimulates the reactive oxygen species that are the waste products of several cellular processes to build up. Overproduction of ROS can lead to several effects, such as peroxidation of lipids, DNA damage, and consequently, apoptosis. So they checked the ROS levels. They checked via fluorescence that there were more reactive oxygen species in the nano-ppam set than the control. Pretty, ain't it? They then looked at how murderous the ppam was at various concentrations, including using 0 micrograms per milliliter as a control, and at TBHP, which is a really good oxidizing agent, aka something that is well known to create reactive oxygen species. They used the ppam and the TBHP on cells with and without NHC, an ROS inhibitor. As you can see, when the concentration goes up, that blue bar dies more readily, whereas that green bar only dies a little. The TBHP shows what ROS can really do, with most of the cells without the inhibitor succumbing to the ROS, and most of the ones with the inhibitor just kind of hanging out. So in this picture, the blue and the green mean different things than the last one. The blue is the normal nano-ppam, while the green is nano-psimp, which is the silica nanoparticle without the pores in it. You can see that the porous one in blue killed a whole bunch more than the non-porous one at every level. And this one is of them checking the ROS levels of ppam, psimp, and that TBHP super good oxidizer versus normal cells. The intracellular ROS level is significantly higher, approximately 3.4 times, in the nano-ppam group, yet we did not observe any significant changes to ROS level in the cells treated with the nano-psimp variant relative to the untreated group. Keep in mind that this is the amount of change in ROS from a normal cell. So the nano-psimp is less than 1-fold difference from normal. The nano-ppam is an average of 3 to 4-fold difference, and the TBHP is an average of 4 to 5-fold difference. The nano-ppam's first standard deviation overlapping the TBHP is super exciting because that TBHP is such a good creator of ROS. Next, they looked at the mechanism not for uptake, but for the killing of the cancer. Generally, cells can die either by necrosis or apoptosis. Necrosis refers to accidental cell death resulting from cellular trauma with loss of plasma membrane integrity and rapid release of intracellular content, which is what's happening to the amoeba in my avatar. We'll have to talk about that story at some point. But yeah, the poor amoeba is undergoing necrosis as its cytoplasm pours out through a hole in its membrane. Conversely, apoptosis is a naturally occurring programmed cell death that is characterized by the process of autonomous cellular dismantling. ROS has been implicated in both modes of cell death. So the question becomes, which mode of death is our little Trojan horse inflicting upon those cancer cells? Creating so much trauma that the cytoplasm escapes, or cellular dismantling? That's what this next section is trying to figure out. So in this experiment, they use annexin 5, which is the stain that gives that gorgeous green color, and PI, the stain that causes that vivid red. Annexin 5 binds to phosphatidylserine, which is a component of the cell membrane that signals apoptosis. And PI binds to DNA, but it can't get through that cellular membrane, so it can only bind to DNA and shine if that cell membrane has already popped. In the control, there were a few cells that lit up in that bottom corner, but for the most part, no signal. In the nano-PPAM, there are quite a few little dots, meaning that there are quite a few cells that were apoptotic. This is confirmed by the next figure, which shows the controls in X and V levels being on average a bit below this horizontal line, then getting shifted up to the line with PPAM. Same with the PI on the vertical axis. This is saying that there is indeed some programmed cell death involved. It's not just some accidental skewering of cells during the method. Alrighty, now that we know that it's apoptosis, we can figure out which kind of apoptosis. You got two possible pathways, intrinsic and extrinsic. The intrinsic pathway is activated by intracellular stress signals, while the extrinsic pathway is triggered by the coupling of extracellular death ligands to the cell surface death receptors. So they look at how much expression there is, and then they normalize it, which just means that they divide it by the amount that a normal cell had. This way, 1 just means the same as the control. CASP3, which is involved in both pathways, was about 2.5 times higher than normal. And that was negated by that ROS inhibitor NAC. The CASP6, which is activated by CASP3 and thus is also involved in both pathways, wasn't really much affected at all, with or without the NAC. CASP8, a major caspase in the extrinsic pathway, was at like 3.5 times normal, which was also brought down a little by the NAC. CASP9, a major intrinsic caspase, was more than 2 times higher than normal, which was brought all the way down by the NAC. And CASP12, which I believe is part of the extrinsic pathway because it deals with cell surface receptors that talk to the outside world, but nobody seemed to want to say the word extrinsic next to, is in a similar boat of being about 2.5 times higher than normal and then being brought all the way back by NAC. They wanted to check their CASP8 results with flow cytometry, so that's where this one comes in. Yay, they're different! They then checked to see if CASP8 was the majority of the mechanism by inhibiting it. That's a common theme, at least in biology. If you want to know if something's the mechanism, knock it out and see if it still works. If it doesn't work anymore, congrats, that's your mechanism. In this case, however, knocking out the CASP8 only made it 15% less effective, meaning that it's not just working through CASP8. It works on several caspases to get apoptosis to happen. Now that they've got that mechanism, they're going to make the cancers look a little bit more like they would inside a body without being in a body yet. In an actual body, it's not just going to be a thin line of cells on a plate. It's going to be a whole tumor in all its lumpy 3D glory. So they grew the cancer cells on a special plate that's designed to allow them to grow in a vaguely spherical shape, and did the tests again. This time they're back to having that control group be the particles with just those naked amine groups on it. After day 1 of PPAM murder time, there was a little bit of a difference between the control and the PPAM. In fact, it looks like the control group's tumors actually grew a little bit. See, it's over 1. Day 2 getting a little better, and day 3 getting really up in there while the control just kind of sits there. They ended up having about a 38% suppression of tumor growth. This picture is of the same cells and got very similar results, but the y-axis is now cell viability. So instead of how much it shrunk the tumor, we're back to looking at how murdery the PPAM got. By the end of the 3rd day, about 40% of the cells were dead, which doesn't sound like that much compared to 80% in 1 day of the 2D cells. But this is why they're testing it like this. The 3D closely packed structure of the cancer will interfere with how any drug can get in there. So 40% over 3 days could be really good. I kind of wish they had kept going on this one just so that we could see when all the tumor cells died, so that we could get a general timeline on how long this would take if done clinically. But there's already a whole bunch of different experiments in this paper. Perhaps a future study, now that we know that it has potential? Okay, so they've already done a whole bunch of experiments at this point. figuring out which amino acid would be best, sizing the nanoparticle, making sure that the ROS got induced, figuring out the mechanism, and now trying it on a 3D tumor. They've basically exhausted what you can do outside of a body, so now they won't waste any mice on things that definitely won't work. So they did the main test. They grafted tumor cells onto the flanks of 15 mice and then treated them. 5 got PBS, 5 got the naked amine particle, and 5 got the PPAM. PBS, the red line, is basically the placebo. The control group has to be treated the exact same way as the experimental group to account for the stress being released by handling or by administration of the treatment. So if you're injecting your Trojan horse into the mouse, you're also injecting your control, just with PBS, which is a type of saline. We already know about our blue line, the naked amine particles. That's the nanoparticle with the amine groups on it, but not the amino acid. It's making sure nothing happens without the actual treatment on it. And look! The error bars of the naked amine protein massively overlap with the PBS. It's pretty normal to have the lines skewed to one side or the other like this when there's such a small sample size. If you test more and more mice, then some will be at the higher end of the spectrum and some at the lower. But when you only test 5, they may all happen to be on one side of the spectrum like this. Then we have our green group, the Nanoparticles. Here there be no error bar overlap, which tells us that the tumors all ended up much smaller, which is verified by the end result examples they gave. They made sure that the mice's weight stayed pretty constant. I'm assuming that this is to make sure that weight wasn't an overall variable, but also to check if it had any gastrointestinal side effects, like nausea. That is speculation, though. For the 2 treatments that used the silica nanoparticles, they used the fact that silica isn't really a thing that's usually found in the body to track where the nanoparticles ended up by the end of the 14 days. They prepped the tumor, the kidney and liver, which are where things get removed from your system, and the lungs and heart, which are where you presumably don't want to have silicon. They found in the non-amino acid treatment that it was mostly in the lungs and the tumor, but there was some in the liver and a little bit in the kidneys and heart. In the PPAM round, it was holy moly, so much in the tumor. And then the rest of the organs followed the same pattern as the amino particles. Finding a good chunk of the silicon in the lungs was a bit worrisome, so they checked the tissues to see if there was any damage. To check if the silicon had damaged anything, they H&E stained the tissues and then looked at them. H&E staining is a super common histology test. They use hematoxylin to stain a bunch of stuff dark purple, including several organelles, several connective tissues found in the extracellular matrix, and DNA. At the same time, eosin will stain a bunch of proteins and muscle fibers pink. I'm not a trained histologist. It would be nice though. But yeah, I'm not a trained histologist, so my opinion is inexpert. But I am a pro at the Highlights magazine Spot the Difference puzzles. So I'd say that heart, liver, and lung look almost identical to me. The kidney looks a little bit different, but not to an extreme extent. Which I think makes sense because silicon dioxide is removed from your system via the kidneys. And the tumor looks a lot better in the PPAM treatment than the other 2. 1 second. Then they looked to see how much apoptosis was going on up in here. Tunnel analysis highlights apoptotic cells in green. There's very little in the PBS treated ones, a few in the naked amine ones, and bam, look at all that green in the nano-PPAM'd ones. Oh. Bye, Elliot. When they quantified it, there was less than 10% cells murdered in both the PBS and the naked amine groups. And we're back to a whopping 80% cells apoptosed in the nano-PPAM group. I do wonder why it looked more like the 2D model than the 3D, but I guess that's why we test things in vivo. Holy crap. One thing I like to do when going through a paper is to moment to see if there's any other way to read the data. Maybe I'm just not being creative today, but I don't really see any other things that this data could mean. If you spot a potential confounding factor that they didn't test for, let everybody know in the comments. So yeah, most of the problems that I had with this paper were pretty small ones. I wasn't a huge fan that they used mostly breast cancer cells for the human cell trials. With things like chemo, there's a wide variability of efficacy in different cancer types. According to Cancer Research UK, chemo generally completely works on testicular cancer and Hodgkin's lymphoma, while it works less well on breast and bowel cancers and needs to be used with other things. Since there's such a variability in reactions between tumor types, using mainly breast cancer cells will tell us something, but it can't automatically be extrapolated to other tumor types. While they did do that one section with the stomach and skin cancers, I would have loved to see more cancer types represented in the spherical and the mouse trials. Also, in the ones that did look at the stomach and skin cancers, they only used PPAM. I know it was generally an order of effectiveness based on hydrophobicity, so it was probably generalizable to other cancers. But I'd still like to check other cancers to make sure that it wasn't uptaken differently for whatever reason. Ooh. Maybe LAT1 is still upregulated, but some mid-sized amino acid transporter is super upregulated for whatever reason. However, I do realize that this is an initial first of its kind of a study. So it's understandable that they do a few different cancer types that are on hand but are still major problem children in cancer research. Until they've made sure that it's at least a little bit feasible anyway. I'm guessing that if they aren't doing that yet, it's probably an avenue of research that they'll do before human clinical trials. Sample size is a complaint that I'm always going to be putting in this section. Again, this is a super initial test of concept, so you don't want to be giving thousands of lab mice cancer for a treatment that you don't even know works yet. Mice are an important model for research, yes, but they are living beings, so we don't want to cause undue and unnecessary suffering or loss of life. This means that the small sample size isn't a poor reflection on the authors, but just a call that more research needs to be done to verify the results. And there's always a chance with small sample sizes that something happened that is non-replicatable. This research is promising and exciting, but if the PopSci article is saying that cancer is cured, or even just talking in absolutes, it's sensationalizing for clicks. Lastly, I'm personally not convinced that the silicon hanging out in the lungs like that won't eventually cause damage. They had only done the treatment for 14 days, but if it just sits there for a long time, it may start getting in the way. Before it was tried in humans, we'd probably want to do a long-term test on mice to make sure that it doesn't start causing damage later in the process. I'd personally put this in the same paper as the test that looks to see how long the PPAM takes to completely kill the cancer cells that I was asking for earlier. So personally, I take this article as exciting and promising. I hope to see this study replicated and expanded upon in the near future. Unfortunately, in the scientific realm, there's not much funding for replicating experiments. There should be, but there isn't. But if somebody wanted to, say, do the experiment where they tried it on other kinds of cancers, then that would verify the findings at the same time as they expand on the science and move it forward, possibly making it more likely to get grant money approval. This paper was published in a peer-reviewed and highly respected journal, Small. This means that this third-party journal is staking its reputation on the idea that this paper is legit after fully checking it over. So we can generally trust that they didn't go making things up or have any big mess-ups. Still, does the method and analysis make sense for what they were looking at and trying to prove? Do you have any ideas about how to make that method better? What do you think the next steps for research should be? Should they get a bigger sample size of mice? Try it with multiple cancer types? Tinker with the payload? Just go into clinical trials? Or something else? One thing I love about this paper is that it looks at the conventional way of doing things, turns it on its head, and sees if the opposite will still work. Weaponizing and giving the cancer what it wants with a bomb attached, rather than just withholding the nutrients like normal. Are there any things that people do to solve a problem that you think may work better if you take it, flip it on its head, and try a version of the opposite? I'll put these questions down in the comments. Please share your ideas and thoughts on these topics, as well as your own questions and thoughts about the paper. My dream for this channel is to inspire us to think deeply and foster curiosity together. Thank you to Wu et al. For your contributions to cancer research. I'm hoping to see this become a tool in the practitioner's toolkit within the coming decade. Thank you to Paul Aschauer for your camera work and mic wrangling during this series. And to Jennifer Garsteg for our wonderful theme song, Paradigm Shift. We got a Patreon. I'm not going to be doing tier levels yet, but there were a few expenses that popped up. So if you like what I'm doing here, please consider donating. I have my eyes on some fancy lighting, but for now I'd mostly just like to cover the licensing for the stock photos I use each month. The next video will be the announcement for our next article. I'm really excited about it and can't wait to introduce you to it. One of the books visible on my bookshelf is going to be a bit relevant. It's also going to be a check-in video to gauge whether I'm actually presenting at the right level for the people, you know, actually watching. But I'll leave my questions until then. I wish you exciting new discoveries. May your day be wonder-filled. Did I do that right the first time? Together we're going to explore the breadth of the awesome shit that's going out. In the Nano PPAM, there's a whole bunch. They also left some as just the naked and meat. That was right.
That's right.
That was right.
Good job.
Okay. Oh, so silicone is a silica ketone.
Yeah. Etymology.
Cool!
Silicon plus own.
That's cool! So it's a silica in the middle with a double bond. That's very exciting.